So good afternoon, everyone, and thank you for joining the first webinar hosted by Appleton Woods in collaboration with Manchester Biogel. My name is Luke Housley and I'm the Business Development and Marketing Manager here at Appleton Woods. If you're not familiar with Appleton Woods, we're an independent supplier of laboratory consumables, equipment and reagents based in Birmingham. We've got over 50 years experience in servicing the research and laboratory testing industry and are very proud to have added Manchester Biogel's product range to our portfolio. Today's webinar is all about setting up reproducible 3-day assays using the animal-free hydrogels that we'll hear about later. And I'm very pleased to hand over to Professor Aileen Miller, who is the CEO and founder of Manchester Biogel, who's going to tell us all about creating the perfect environment for your cells. Over to you, Aileen.
Hi, my name's Aileen Miller. Thank you for that introduction. So I'm going to tell you a little bit today about setting up reproducible 3D cell culture assays, mainly using animal-free hydrogels called PeptiGels, but I'll introduce what 3D cell culture is, why it's important, and also how to choose your animal-free hydrogel, or why we want to use animal-free systems. So I don't really need to tell you what cell culture is. So very briefly, it's creating an artificial environment in the lab where you're using biological cells and you're allowing them to grow and interact with each other and also their surroundings in either 2 or 3 dimensions. And typically people do this for various reasons, tissue regeneration, cell biological fundamental studies, or trying to develop human or disease tissue systems. So typically, you're using cell culture well plates or inserts within the well plates, or you're bioprinting using the hydrogels as an ink to print your cells. So why is this actually important? Why do we want to put so much time and resource and effort into all this? Well, typically we've got a whole range of different applications here, ranging from fundamental cell research, where you're looking at trying to understand cell biological pathways for understanding cancer behavior and reaction to cancer drugs, for example, through to vaccine manufacture, so how you can actually manufacture live vaccines on a large scale and how they actually effective are those vaccines, very prevalent in today's society, all the way through to sort of regenerative medicine, tissue engineering, so looking to replace body parts or help liver transplantations and such like, all the way through to building disease and organ models, and that's more complex culture systems, and you want to do that partly to understand more about the disease and more about how organs function, but also then thinking about transplantation models, and then also then leading on to testing new drug modalities, so really pushing into drug discovery, looking at how effective and how efficacious your new drugs are they toxic, so understanding that process. And if we can do that using cell culture models throughout, then we're actually increasing the reducing the time it takes to get to market and reducing the cost of taking new drugs and regenerative solutions into clinical practice. So these are just some of the areas that are used where 3D systems are used. It's a huge market area. It's worth about $2 billion today, and it's got a CAGR, which is a cumulative aggregate growth rate. So this 2 billion is growing 20% on average year on year, and that's predicted to grow all the way up to about 2030 now. So very economically important area as well. So these are just, again, just a range of different application areas, broken down a little bit more, all the way from, looking at organoid growth, we'll talk a little bit about that later, drug discovery, toxicity testing, wound repair, printing, also used in personal care, so all of your creams and such like are tested in vitro systems, and they want to get that right and get 3D systems all the way through to your dental regenerative medicine, also drug delivery applications that we've kind of touched on. Okay, so we talked about some of the application areas in these systems. So where is the technology up to today? So 2D cell culture is very well known. That's where you take your cells and you put them down onto a surface. Typically, you're putting them down onto tissue culture plastic, so flat surface, hard surface as well. Very well established, these methodologies, and it's currently the go-to place to start, predominantly because the results you get are simple to analyze, simple to visualize, and because it's just you're looking at one layer. So you put your cells down, they grow in these 2 dimensions. The cells typically then interact with each other, have very limited interactions with a very hard tissue, typically tissue culture plastic surface. So the data that you get in the behaviour of the cells are not representative of the in vivo environment where you have your 3D supportive and natural extracellular matrix. So that's important because it's not only not representative, that means that your cells don't behave in the same way. They don't have the same cell-to-cell interactions. They have altered signalling pathways, which then gives different cell behaviour. So you don't tend to then get the same predictivity when you're looking at new drug testing, whether it be for how active the component is or how toxic potentially the component is. So you then need to typically then use go for animal testing for validation. It's also quite limited in its applications because you can never generate a new organ, for example, if you're only going to be using and growing cells in 2 dimensions. So we much prefer, and the community is recognising that we need to move from 2D to 3D. Of course, 3D cell culture, it's not as widely explored, so not all the protocols are there and readily used. So that's where we need to put some R&D efforts into making sure that they're widely applicable. You can do all the tests with them. And to really demonstrate that not only do you get more in vivo environment replicated behaviour, but actually the results and the cells behave as if they're much more in an in vivo type environment. So you get enhanced cell-to-cell interactions and signalling, and actually, things like stem cells in 2D, you don't always see the right phenotype forming, but in 3D, where you're really representing in vivo type behavior, you can get the correct phenotype and direct the phenotype behavior much more closely and much greater control. So you get much more reliable and relevant, physiologically relevant results. So it gives you an improved method to model diseases, also model healthy tissues, and also then build up more complex structures that are reminiscent of in vivo tissues, disease tissues and models. So then you can think about your transplantations we've touched on. Okay, so those 3D systems are great. We've all agreed now that 3D is the way forward. So there are various ways to do 3D cell culture, and the main one is where you use a scaffold, typically a hydrogel scaffold, for your cells to go into, cling onto, move around within, and feel that supportive environment, just like they would do in a natural extracellular matrix. So how do we choose which scaffold to use? So there's a whole plethora of materials out there in the literature and available commercially. So which one do you go to? So we have categorized them into 2 camps, well, not just we. This is well known. So there's the natural based systems, which are your typical polysaccharides and your proteins. So of course they're available in nature. So here there's general pros and cons with these types of materials. So these include things like cellulose, collagen, proteins, also alginates, etc. So there's various pros and cons. So with these types of materials, they're inherently biocompatible because they're natural materials. They're also inherently functional in some cases, so they contain some bioactive signaling sequences. However, because they're natural materials, you can't control the molecular structure or material properties and behaviour. So they often come with huge batch-to-batch variability. They're not often or always chemically defined. which leads also then to greater batch-to-batch variation. Also, their production is not scalable because you're using natural resources, and some are abundant and some are less so. Typically, because they're natural, they're also not clinically translatable, and typically it's quite difficult to tune the material, whether you want additional functionality or want to tune mechanical strength. So the other side, we have, before we talk about that, I'll talk about the most commonly used natural material, and that's PeptiGel. So PeptiGel actually accounts for 70% of today's market in terms of scaffold use within the R&D community. Now, PeptiGel, if you don't know, it's a tumour that is derived from mouse sarcoma cells. These are injected into the mouse. The tumour is allowed to grow within the mouse, just as in this one on the left-hand side here. So here, the mouse is sacrificed, the tumour is extracted, is decellularized, and then sold directly as PeptiGel. So it takes around 3 mice, we've estimated, to make every 10 mils of PeptiGel. So to meet today's current market demand, it's over 3 million mice are required to be sacrificed every year just to make PeptiGel. So that's not particularly great. But because it's an animal-derived product, that also means you're never going to be able to translate it into clinical applications. And also it comes with huge batch-to-batch variability because, of course, every mouse is different and grows the tumour differently. So the batch-to-batch variability is really well known. And also, you don't know the product composition. PeptiGel comes with a whole soup of growth factors that help it do its job, but nobody knows what it is exactly in the PeptiGel that is doing or eliciting specific cell responses. And often you don't get the right physiological or human physiological response that you're looking for. They're also not tunable. They're a little bit of a difficult to handle, a bit of a faff to use. You've got to put them on ice and then bring them back up to room temperature. So it can be time consuming and just a bit of a pain to set up your experiments. Also slightly coloured, so it's not great for optical and fluorescent microscopy. So that's the natural systems. We can now go on and talk about the other side, which is the synthetic materials. So this covers things like your polymers, a lot of ceramics, metals, which are harder surfaces and systems. So with these types of materials, you get typically your polymer hydrogels. They also come with pros and cons. So cons, they have limited biocompatibility because you're synthetically making these systems. They're typically not functional, or you've got to do extra steps to get the functionality embedded within them. And typically, certainly with the polymers, you need to use organic solvents to make them, which is not environmentally friendly. However, because they're synthetic, you can make them reproducibly, fully defined, fully scalable. We'll know how to make tons of these materials typically. And they're quite often cost effective, and because they're synthetic, you don't have the ethical issues that you have with, for example, PeptiGel. There is, however, a third class of materials, and they are PeptiGels, or peptide hydrogels, and they typically combine the pros of both the natural and the synthetic materials. So because they're made from short oligopeptides, so each building block is made up of several different amino acids. Amino acids are what make up proteins in the body. So they're inherently natural-based systems, so they're biocompatible. You can choose the amino acids to make the peptides functional, so bioactive. They are made using synthetic processes, so they can be made fully reproducible, defined, and we can make them at scale. And it's now, over the past 20 years, the cost of making these systems has gone down considerably. So they're now cost effective, and of course they're synthetic, non-animal derived, so they're ethical. So we believe that peptide hydrogels are the way forward and offer superior material for using as a biomaterial scaffold for your 3D cell culture. So let's explore these in a little bit more detail. So here we've got a video just showing how these systems sort of self-assemble, and these are sort of our peptide hydrogel materials. The different coloured balls that you see are amino acids with different properties, so they can be negative, they can be positive charged, they can be hydrophilic, they can be hydrophobic, combined in specific ways to create your one single peptide, and then these are defined to form your beta sheet self-assembling fibres, so they self-assemble in one direction, and then these fibres entangle, they associate, they feel each other just like spaghetti on a plate to form your three-dimensional hydrogel scaffold that is self-supportive and transparent, as you can see in the image there. So our systems from Manchester Biogel are all tunable, so you can replicate all human tissues in terms of their functionality and their mechanical strength, and they were developed out of many years of research from the University of Manchester. So a little bit more about them. So I've talked about some of these unique selling points, USPs, previously. As I said, I cannot stress enough that they're animal free. So they're nice and ethical and can be used and translate research into the clinic. The key thing really is that they're reproducible, so you get reproducible and reliable data every time that you use them. And they're inherently biocompatible. We do supply them ready to use. So as you'll see shortly when we look at some hints and tips on how to use these systems for your setting up your assays, you just mix in your cells nice and easily at room temperature, and then you plate them out or print them so they're ready to use. Transparent is important because you want to be able to use them, you want to be able to see your cells, what the cells are doing and how the cells are behaving. So they're compatible with optical, fluorescent, confocal, etc. microscopy. And they're printable, so you can use them in liquid handling or 3D bioprinting, but also injectable and sprayable, which allows them to be easily handled in a more clinical environment, but also, of course, easy to handle and use in the lab as well. Okay, so what kind of systems do we have? So we have a whole sort of family of peptide hydrogels. As I said previously, that we can replicate all human tissues. So you've got soft breast tissue, soft brain tissue, all the way through to harder, cardiac tissue or cartilage in your ear, for example. So we can replicate the mechanical strengths of all those types of tissues, and also slightly different charges. So some cells like negative, some like positive, some like neutral. Charges on the surface, just really thinking about the interactions with the surface. So we have a whole range of charges, and we can help direct you on our many years of experience in terms of what hydrogel to use for your cell type. We do recommend when you start just to try our starter pack, which contains 2 mils or 5 mils of each of our 5 key hydrogels that we typically sell the most of, our customers use most of all, alpha 1, 2 and 4, and then gamma 2 and alpha 2 RGD. So it just gives you a feel for a range of hydrogels with different mechanical strengths and different charges, and allows you to try out to find the best peptide hydrogel for your cells needs. We've also got functional kits. So here we look at introducing functionality or bioactive signals into our hydrogel. So here we've just got some examples where you can introduce the functionality of fibronectin, collagen, and laminin. For example, the top example is where we've got fibroblast cells, and they're introduced into our alpha 1 PeptiGel, and here we've got alpha 1 that's included some RGD on the system. So the cells survive and grow in the vanilla, the plain gels, but sometimes you just want to expedite that process, and that's what the RGD has done in this case. We can also introduce full laminin sequences, and this is what we've done here on the bottom with iPSCs. So here they're clumping a little bit in the vanilla hydrogels, and we introduce a small quantity of laminin. Can be animal-derived or recombinant laminin. We can see that the iPSC cells are spreading out and forming a nice sheet-like morphology, as we wanted them to do in this case. So what I'm going to do now is just show you a few example studies, just to give you kind of a feel for how the hydrogels are used, and sort of stimulate your interest and also stimulate your questions, really, in terms of how you want to get the best out the materials that you're using, and that we're here to help. So if you have any questions, please feel to pop them in the chat if you haven't, or save them up at the end, and we're here for some time to help you with your own research. So this is our first example. It's looking at stem cells. In this case, it was mesenchymal stem cells. And this work was done by a student who was a dentist by training. So he was using MSCs to differentiate into osteoblasts to then recreate and get bone generation. So here we're using non-stimulated media and stimulated media, and here we've got the cells embedded in PeptiGel Alpha 1. And you can see from the staining that in stimulated environment, when we have taken the MSCs and converted them into osteoblasts, then we're getting the hydroxyapatite starting to form and be deposited, which is the start of the bone formation. So this work is ongoing and looking to create a regenerative treatment for sort of bone disease, and in this case, dental repair. This is another example where we're using the sprayability and the injectability of the hydrogels. So this is thinking about wound healing. So here in this example here, there's a wound, an artificial wound created in the skin. Here we have taken the hydrogel with keratinocytes, injected it just below the surface or just in the wound site, and you can see that the keratinocytes are growing nicely in the alpha 2 PeptiGel this time, and you've got a nice smooth layer on top where the cells have formed a epithelial layer and remodeled the top of the epidermis. So here we're using the hydrogels to regenerate skin. We're using these really to start to develop models for cosmetic testing. This is an example where we've got a co-culture system. So here we're creating and combining the 2D and 3D aspects. So here we're using PeptiGel Alpha 5. We've got fibroblasts mixed in 3 dimensions, and then we've got oesophageal cells on top in 2 dimensions. And you can see that in each case we've got the fibroblasts growing nicely. We've got the forming a nice functional network underneath, and then on the top with the epithelial cells are forming nice epidermis system. Here you can see you've got a nice mottled structure, which is what you would expect, and you're getting a nice uninterrupted epithelial sheet forming there. So this is looking at creating a sprayable solution to go onto the surface of your throat to treat Barrett's esophagus in this case. Now we're going on to a 3D tumour model. So in this case we looked at, last example was 2 different cell types. Here we're combining 3 different cell types. So here we're taking cancer cells, mesenchymal stem cells and HUVEC cells, which form vascularization within the structure. So we're trying to recreate a vascularized tumour model that can then be used for drug testing of new therapeutic entities. So we have used PeptiGel Alpha 1 and 2 in this case, and combined each of these cells into one culture. We have done them individually and demonstrated the work, and then we've taken all 3 together. And as you can see, this is the cancer organoids, and they're forming nice clusters as we would expect, and they're expressing the right cell signaling sequences. And then here we've added the HUVEC cells to the system, and we've got the vascularization within the 3-dimensional tumour. This is work that is just been submitted for publication from Helen and Olga in Manchester. We've also, thinking along the theme of organoids, we've also done kidney organoids. This is an example from Prof. John Crean's group at University College Dublin by his PhD student Niall. And here we've taken iPSCs and directed them to the renal cell fate, and these are just cross sections where we have looked at the staining of the kidney organoids, and we're demonstrating that the organoids are the right size, the right shape, and also they're now stained and looked at the cell markers, and they're supporting the differentiation into kidney organoids, and that work is progressing, and we have an update on that next week, so I'm excited to see what's happening in that area. The last example is actually just taking the 3D cell culture that step further and thinking about creating 3D bioprinted structures where we can increase sort of the complexity, potentially use different materials to this great spatial construction and intricate model systems. In this case, we're looking at 3D printing of breast tumour cells, in particular mammary epithelial cells. We're using alpha 1 and alpha 4 in this case, and here you can see a printed structure. This is just a hash structure. It was one of the first ones that we did with Marco Dominguez's group in Manchester. And here you can see that in the straight parts that the cells are stained, in this case for alive dead. So the cells that are alive, there's a few cells dead just at the edges and even in the corners, and that's just due to the pressure as it comes out the end of the needle. But the majority of the cells that are alive, and they've gone on to last for several weeks in cell culture, and then they're expressing the right cell markers. So we're working on, and many customers are looking at different structures for different applications, and this is just an example of printing our Manchester Biogel V logo. So what I've hoped to have kind of shown in that very brief introduction to such 3D cell culture, 3D biomaterials, is that PeptiGels, or peptide hydrogels in general, offer superior scaffolds to any of the sort of traditional natural materials or the traditional synthetic materials that are used. And they open the door to lots of different application areas and lots of different cell types. I gave you a few examples today. We've got many more moving forward, and many more as more customers actually use and adopt the hydrogels in their workflow. So I will leave it there. Feel free to contact us on any of our social media channels. We're on Twitter, Instagram, LinkedIn. And have a look at our website for further details. We have got a lot of the protocols that Dami is now going to show you and share some hints and tips with. They're also on our website along with some frequently asked questions. But please do keep asking your questions in the chat, and we will get to them at the end. Yeah, please interact. You're here to learn, and we're here to try to help you. So thank you very much. I will pass on now to Dami, where he'll show you a little bit about the cell culture assay setup.
PeptiGels are synthetic hydrogels that can be used within 3D cell culture models, bioprinting and drug discovery. Their ease of use means they can be taken straight out of the fridge and to your bench to start your experiments. There is no requirement to defrost on ice, and therefore no time is wasted. Simply centrifuge the PeptiGel to remove any air bubbles, and you're all set to begin. PeptiGels can be pipetted directly into a cell culture well, or alternatively into a permeable insert. If you are using an insert, pre-wet the membrane by either pipetting PBS or cell culture media. Taking an empty tube, pipette the volume of PeptiGel you require carefully, ensuring not to introduce any air bubbles. For this step, you can use either a positive displacement pipetter or a 1 ml single channel pipetter. Next, take your tube containing your cell suspension and using a pipetter, aliquot the volume of cells needed into the tube containing the PeptiGel. Pipette the cell mixture slowly whilst moving the pipette tip in a circular motion to ensure a thorough mix and uniform dispersion of the cells, again taking care not to introduce any air bubbles into your cell gel mixture. Then take the required amount of the cell gel mixture and, depending on what your protocol dictates, pipette into either your prepared permeable insert or directly into your cell culture well. Next, take your cell culture media and add to the individual cell culture well where you have pipetted your hydrogel. Or to your insert and the surrounding well. When adding the media, take care to pipette down the side of the well wall, ensuring you do not disturb your cell gel mixture. Replace the lid to your plate and then incubate for 30 minutes at 37 degrees. After incubation, aspirate off the media and discard, making sure not to disturb the gel with the pipette tip. As you can see, a uniform cell sphere has been created. Replace with fresh media and incubate either overnight or for as long as your experimental protocol requires. One of the advantages of using PeptiGels is that they are transparent. Therefore, your cells can easily be visualized under a microscope, and if using fluorescently labeled cells, these too can be viewed with an appropriate fluorescence microscope.
So hopefully Aileen's presentation and video showed you there how the PeptiGels can benefit your work. And as I mentioned earlier, we're very pleased to have partnered with Manchester Biogel, and we can offer a complete solution for your 3D cell culture. We're both small businesses, and hopefully we can be flexible and respond quickly to your specific requirements. If there's any opportunity, there'll be an opportunity, later on to answer any questions you have. So hopefully that can help you achieve some consistent results. The next slide shows some of the products we can supply in combination with the PeptiGels, which you may have seen in Dami's instructional video. This includes everything from tissue culture plastics, such as the permeable supports you may have seen, to microscopes and any other pieces of equipment you may need. And on the next slide, you'll see some contact details to get in touch with us. So any more details that you require, both on the plastics, the equipment, or the PeptiGels, please don't hesitate to get in touch. I'll hand over now to the Manchester Biogel team to answer any questions that we've got in the chat. Jess, I don't know if you can share those.
Yeah, hi everyone. So I'm Jess, I'm the Business Development Manager at Manchester Biogel. And now it's time for the Q&A session. So let's have a look at the chat. If you've got any questions, please feel free to write them now. But in the meantime, we have actually been emailed some questions ahead of the session today. So while we're waiting for you all to write your messages and questions, I'm just going to read out some that we've been asked already. The first one being, how do you ensure the materials you use give the same results every time? So because PeptiGels are fully synthetic, it means that we can control how they're made. So there's no batch-to-batch variability at all, which means that you get your reliable and reproducible results every time. The next one is, how do you incorporate functionality within the peptide hydrogels, and how do you control their concentrations? Oh, okay, so I'll just read this answer and then I'll go into the ones on the chat. So the functionality is incorporated into the peptide hydrogels with the addition of protein mimetics tagged to the end of the self-assembling peptides. We control the concentration at the production stage and can vary these based on customer requests if desired. And the functionality can also be done manually by the customers through the addition of protein factors into the culture medium before adding to the hydrogels. So that's a couple of ways in which we can functionalise them. So now on to the questions. We've got a question from Manohar that asks, what kind of preliminary assay can be performed to assess which version, type of PeptiGel among the 5 subtypes shall we use? So Aileen and Jamie?
So basically, when you buying the starter pack, the first assay we recommend is to look at the morphological characteristics of your cell. So and compare that to the in vivo phenotype. So if, for example, your cells have displayed like a cuboidal-like appearance, or say maybe like a fibroblastic appearance within the natural in vivo environment, then you can see if you can replicate that phenotype using PeptiGels. So out of those 5 PeptiGels, one of them will probably give you the best phenotype.
Okay. The next one from Laura Randall is, have these gels been used with precision cut tissue slices?
Some of the studies that have used primary cells, sometimes they've had to like cut some of those slices, and not particularly maybe embedded into the gels directly, but some have then had to like digest the tissues and then extract primary cells into those the gels and grow them. But if you're thinking of maybe like I don't know, for histological based kind of characterization where you want to cut the slices and put directly into the gel, there's a possibility that that can work as well.
And the next one is from Maria Esposito, and she said she's interested in leukemia models. Has anybody tried to use these biogels to recapitulate the bone marrow?
So at the moment we have there's some preliminary studies where people are beginning to extend this into blood-related kind of studies, particularly beginning to look at migration ones. And because they are applicable both for adherent and suspension cells, so they can be used for leukemia-based kind of models.
Okay. And there's another one from Hannah Smith. How can these gels be used for neurodegenerative diseases specifically to produce a specific environment applicable to brain tissue?
So we've got some case studies, particularly looking at the use of PeptiGels in neurodegeneration. And one of the studies actually look at the use of mesenchymal stem cell, ADMSs particularly. And that's where they've used as a platform to differentiate those cells into neuronal-like cells. And we've also done a couple of tissue-specific experiments to show proof of principle that it actually works. And also in terms of producing the specific environment applicable to brain tissue, one of the first things we look at first is the mechanical stiffness of the brain. I'm sure if you followed on from the presentation given by Aileen, you can see where she was talking about being able to fine-tune the mechanical properties to match what you have within a particular tissue. So once we've been able to match that to the brain tissue, then we can supply the functionalized one. But if you're interested, we can forward a couple of case studies within that particular aspect of the work to you.
And just to add to that, Danny, we have done a few things with looking at delivering to the brain. So you can inject the hydrogels, so the shear thinning, that's why you can sort of 3D bioprint with them. So you can actually also inject and spray the systems. And there is one project that I am aware of that is looking at developing treatments for stroke, for example. So they're injecting the hydrogel into sort of the damaged tissue area of the brain that occurs after stroke. So you can actually deliver it whether you want to deliver it, the hydrogel on its own to act as a sort of a biocompatible structuring material, or whether you want to deliver cells so you start to sort of regenerate and repair nerving things, etc. That's up to, I guess, the sort of the application scientist and direction. But you can deliver it as well as mimic the brain tissue.
Okay. And the next question is from Teresa Tropia, and she has asked, what is the average amount of cells required for seeding and performing a 3D core culture? What is the chance that two different cell culture media can mix?
So for the average amount of cells, normally the protocol has been designed for between 50 to 100,000 cells per mil, up until like 1 million cells per mil. The choice of the amount of cells is dependent on your experimental setup and the proliferation rate of your cells. So if, for example, your cells doubles within, let's say, 18 to 24 hours, which is very common with cell lines, and depending on what your intended outcome is, then that can determine how much cells you have to like plate. In terms of performing a 3D co-culture, what is the chance of two different cell culture media can mix? So if you use a cell insert, and like Luke was saying earlier, may be able to advise on the best one to use actually, or if you speak to myself as well, then you can have a different media at the top compartment and another different media at the surrounding walls of the insert. In terms of the cell culture media mixing together will then determine on what you actually want to achieve, and also what's the influence of the two cell culture media mixing together, cell behavior naturally.
Okay, thank you, Dami and Eileen, for that. I think that, well, we've come to the end of the questions anyway, and we've come to the end of the webinar. Good timing. So thank you everybody for your time today. And get in touch with us or Appleton Woods at any time.
Yeah, thanks everybody for listening, and yeah, feel free to get in touch with Appleton Woods or ourselves, and we can, in particular, Manchester Biogel can help with the technical aspects and applications of the hydrogel. And Appleton Woods have got a supply of the materials. So feel free to get in touch with either of us, and we'd be happy to help and answer questions. I think we're all both on all social media channels as well. So feel free to follow us for up-to-date news and promotions, etc. Thanks very much, and hope to meet some of you soon.
Thanks everyone. Take care.
Thank you.